All right, well, welcome everyone. As people are still arriving, maybe I'll just start by introducing our webinar today. Welcome to the International Soil Carbon Network, AGU and USDA Climate Hub sponsored webinar series towards a durable understanding of soil carbon as a tool for climate adaptation and mitigation. This month, we're going to focus on a broad question: when does increased soil carbon storage yield net removal of greenhouse gases? And we're going to address this at a broad conceptual level, but also think specifically about those high global warming potential greenhouse gases, in particular N2O, and consider how changes in soil carbon stocks may or may not yield the climate benefits that we think they might at face value until we account for the emissions of those greenhouse gases. And we'll consider ways to reduce the emissions of those gases and also ways in which soil carbon and those gases may create feedbacks that are unwanted and how to mitigate those feedbacks. Joining us today, we have two fantastic speakers. I'm really excited about what we're going to hear today. Our first speaker will be Claire Chenu, joining us from France. Some time zones ahead, and many thanks for that, Claire, for joining late in your day as it is. Claire is the research director at INRAE, which is France's National Institute for Agronomic Research. She's also a professor of soil science at Agro-Paris Tech and the vice chair of the scientific and technical committee of the 4 per Mil initiative. The second speaker we'll have today, midway through the webinar, is Jane Johnson. She's a scientist and research leader at the USDA-ARS in Morris, Minnesota. She's a fellow of the American Society of Agronomy, and she's a US co-chair of the Global Research Alliance, an international body that is promoting research to reduce ag emissions. And with that, I think we'll lead into the first talk. Claire Chenu will begin addressing the question: when does increased soil carbon storage yield net removal of greenhouse gases? And take it away, Claire.
Thank you. So good afternoon, everyone. So we were just discussing that this landscape could look like Northern California. Well, it's actually a landscape from Southwest France, and I'm talking to you from Paris area. I live in Versailles. So I'm very pleased to be with you today. And actually, I am someone working in France at INRAE on carbon dynamics, soil carbon dynamics in agricultural soils, and on carbon storage in agricultural soils. So this is basically what I'm working on. And well, you know that in the different components of the carbon cycle at the local scale can affect soil organic carbon storage, that is the increase or the decrease, the increase of stocks, and the increased carbon stocks come from a positive balance between inputs and outputs, and that the current state of knowledge shows that what is most efficient is to increase organic carbon inputs to soils compared to trying to reduce outputs, and these primarily through enhanced biomass production by the agroecosystem. And if we consider this with time, so let's have a very simple graph where you have these organic carbon stocks as a function of time. Let's set a baseline. This would be actually the baseline of average evolution of organic carbon stocks in mainland France, a very slow, low slope decrease, and it's more or less the same across Europe in average. So this is the baseline. Now, if we implement a carbon storing management option, for example, implementing cover crops in the rotation, the carbon stocks will increase and will somehow plateau, reach to a new equilibrium. Storage is limited in soils. If another and this defines the carbon storage potential. So the carbon storage potential is the maximum gain in carbon reachable for a given timeline. For example, my green arrow is located at 40 years. under a given land use and management option. It depends on climate and on soil type. There's no general carbon storage potential. It is context dependent. And if another management option is applied, let's consider, for example, agroforestry, maybe the new equilibrium will be higher. What you can also see on this graph is that storage is low. It takes years, decades, that it is reversible if the management option is reversed to the initial one, and that what is most detrimental is changes in land use, for example, returning permanent grassland. The loss of carbon is then rapid and huge. So this is for carbon, well, the general feature. Now, the question here is net removal of greenhouse gases. So a balance needs to be made, a balance between the fluxes of non-CO2 gases, methane and N2O, and the balance between variations of above ground carbon stocks in above ground biomass, litter, roots, and soil, all expressed in the same unit, CO2 equivalents, considering the global warming potential of the different gases, CO2 being the reference. Now, I would like to I have found that if one wants to, the carbon storage, increase stock storage measurements or increase stock storage to yield a net removal of greenhouse gases, there's a need to account for different things. And I will give you examples for the 4 of them. First is accounting for the whole soil profile. And a quite classical example is the effect of no tillage on soil organic carbon stocks. This is a graph from Metaanalysis from Luo et al. That showed very clearly, but there has been a number of meta-analyses and studies that with no tillage, the concentration of organic matter and organic carbon stocks increase in soil upper layers, while they tend to decrease or not to change below the upper layers, so that the balance, and it was the case in the meta-analysis of Luo, can be null. Overall, there are small effects on soil organic carbon stocks if you consider the whole soil profile, and this is pedoclimate dependent. It is especially true, this very small effect, in temperate conditions. So if for the observed carbon storage to correspond to a net offset of greenhouse gas emissions, there is a need to consider the whole soil profile. There is also a need to account for lateral transfers of soil organic matter. And here I am essentially thinking of exogenous organic matter. Let's consider one situation where a given resource of manure is applied by a farmer, distributed between 3 plots, as I shown on this photograph. And one day he decides to change, and he applies the whole manure on only 1 plot. In this plot, the carbon content, the carbon stocks will increase, carbon storage will increase, but the net uptake of CO2 from the atmosphere at the landscape level will not change. Carbon has just been transferred from one place to another. And this, for example, has raised much discussion, evaluations and discussion about organic agriculture. Well, I'm thinking of organic agriculture because it's an agricultural system that relies a lot on the application of manures and composts. Gattinger et al. Showed, for example, that there was indeed an additional carbon storage when comparing organic farming systems with conventional ones, but there was also much more organic matter added as manure, compost, and slurry. And as was discussed and shown by Leifeld and others, in many cases, the external resource, the organic matter resource came from outside of the plot, outside of the farm even. So it's just transferring organic matter through the landscape, not net carbon uptake, so not net carbon sequestration. So organic carbon sequestration has a different meaning that organic carbon storage. It corresponds to the storage in soil that allows for a removal of CO2 from the atmosphere for a long duration. So in my example of moving around manure, there was no additional carbon sequestration, but additional carbon storage in the plot that received all the manure. So accounting for lateral transfers of organic matter. There is also a need, of course, to account for N2O emissions. And here I will use an example of a long-term experiment on which I work, where in rotations with cereals, well, wheat 1 year over 2, we compare since 1998 different alternative cropping systems that are compared to a conventional one, and you see the main characteristics here. So we have a low input system with reduced mineral fertilization, reduced tillage. That leads to slightly reduced yields. We have a conservation agriculture system with no-till, with also reduced mineral fertilizer based on the low input system, with a permanent coverage of the soil by a plant, and it's now alfalfa. And it also has yields that are lower from compared to the conventional, and an organically managed system that receives no manure, no compost. It is actually representative of some systems we observe at Farmers Place in Paris area. There's tillage each year, of course, no mineral fertilization, but there's a wide use of legumes in the rotation and lower yields. Now, we compared the organic carbon stocks at the start of the experiment. We compared them again in 2014. We just remeasured them, but didn't plot the new results. And what we observed is that there was a slight increase of organic carbon stocks in the organic system, but with very low increases, but good, an important increase in the conservation agricultural system. And actually, we were able in this trial to show that the increase could be completely explained by the additional biomass due to the alfalfa. Bénédicte Autrey also measured nitrogen leaching. Well, she estimated nitrogen leaching. You see it doesn't vary much between the different systems. And she measured over 2 years continuously the N2O emissions. And she found that they were much larger in the conservation agriculture system compared to the conventional and to the low input. The amounts of mineral nitrogen added of mineral fertilizers are the same as in the low input system, so it's not that. And the explanation is actually that there would be additional denitrification because of this mulch that remains humid with low C2N alfalfa over the year. So this would create conditions, and the soils are slightly acidic. So this would create conditions for additional N2O emissions in the conservation agricultural system. So this is one case study. Now, if we consider different management options, Genet et al. Assembled different meta-analyses where had been quantified either the additional organic carbon storage or the additional N2O emissions when implementing different management options. It is actually very rare to find meta-analysis that compile the two types of information. So it's usually separate ones. The carbon storage is represented as negative, an uptake of CO2 from the atmosphere, so it's in CO2 equivalent, and the N2O emissions are represented also in CO2 equivalent, but is in green. You can see that N2O emissions can decrease the positive effect, the effect of carbon sequestration, but in some case nearly compensated, like again in assembling different meta-analysis on no tillage. So it is necessary to establish a full carbon balance when implementing new management options in order to estimate what is the net uptake, well, the net balance of greenhouse gases. And it is also very important to consider not only the short term, but also the long term. This paper by Lugato really brought yes, information that is kind of provocative. Well, makes you think. They measured using, well, they modeled, excuse me, using DACENT, so a biogeochemical model. They modeled the effect of the implementation of two different management options on carbon storage and on N2O emissions. And they did that on the nearly 8,000 sites of the LUCAS. So LUCAS is the European Soil Monitoring System. So a wide diversity of pedoclimatic conditions. The two management options they compared to the baseline were integrated crop residue retention with reduced tillage, and we're implementing a cover crop between successive cash crops as soon as there were more than 2 months between two successive crops. And this cover crop was a legume, N-fixing one. What you can see, so the graphs represent carbon storage. So this is here in green, and it is negative uptake, and N2 emissions in red and appear as positive. What you can observe for the integrated crop residue retention is a small effect on carbon stocks that plateaus around 2050, and a small but continuous increase of N2 emissions. At the start, there's low N2 emissions because the nitrogen is assimilated by the microbial biomass that is decomposing the additional residues added to soil. And after it steadily increases, the balance remains in the negative zone. So it's favorable. Now, if you look at the scenario with the N-fixing crop, you see that the carbon storage is much higher and it also plateaus. But N2 emissions, they never plateau. And the result here is in their simulation, after 2060, there's a net emission of greenhouse gases in this system. So this, I think, is really stimulating to devise cropping systems where, well, to think differently the different timescales and to devise cropping systems where N2 emissions will not offset the carbon storage. Well, in their simulation, the mineral fertilization was adjusted to 70%, 75% of the fixed N by the legume. So there was some mineral fertilization. Okay, so think about time. And again, let's go back to the case study of this comparison between alternative agroecological system in cerealiculture. What Bénédicte Dautrey did also is what I showed you already is she quantified organic carbon sequestration. It's here in yellow. She measured direct N2O emissions, but she also quantified through the leached nitrogen, the indirect N2O emissions. They do not vary much among the system. She assessed fuel combustion in the different cropping system and the cost in terms of greenhouse gases of the synthetic fertilizer used. You can see that in the conservation agricultural system, the other greenhouse gas emissions related to the cropping system compensate for the additional carbon stored. So this is kind of disappointing, and it's nearly the same situation in the organic system. This is kind of disappointing to have a nil effect at the end, but if you compare to the conventional and to the low input system, these are net emitters. Here you have zero net emission, and here you have systems that are net emitters. So it's necessary to measure, to establish the greenhouse gas balance between carbon and N2O, to consider the different operations in the cropping system, to integrate different durations, and to consider the long-term effects as well. And the last example and point I would like to raise, where I do not have strong statements to make, but it corresponds to questions that we repeatedly have from farmers in France and from actors of the food chain. They ask, what is best? Should they return to the soil the residues, or the composted residues, or the digested after some methanization, or even the pyrolysates after pyrolysis? And actually, there is nearly no study. I just was able to find one study quantifying the balance of carbon over the biomass process chain. So they measured, Thompson et al., so Danish colleagues, they measured made measurements, so they were able to establish the balance that for one given amount of carbon in the feed, 100%, they applied it to soil also the digested feed. So digested only remained 20% of that. The faces of the animals that had consumed the feed, and this corresponded to 30% of the initial carbon in the biomass, or the digested faces, this corresponded to 16% of the initial carbon in the biomass, because of course there had been losses by respiration and by as methane. And they measured the balance in soil, what remained after 2 years after applying these 120 and 30 and 16% of the initial feed. And they actually found that there was not much differences in terms of soil carbon that were stored. So again, this is the only study I could find. So this is likely to be variable among conditions, but I think this is a very important question that more studies are needed there. So now to conclude, I just have 2 more slides. In fact, when does increased organic carbon storage yield net removal of greenhouse gases? For that, it's all about limits of the system, accounting for the wholesale profile, accounting for lateral transfers of organic matter, accounting for N2O emissions in the system, from the soil and in the cropping system, and accounting for the balance of carbon, nitrogen in the process chain of the biomass. And there are, for that, well, a number of research needs. Well, to give you again one image of what we're doing, I'm coordinating a European joint program, the joint program Soil, that assembles or participate 24 institutes from 24 European countries. And we have different expected impacts, and we have defined the knowledge needs. One of our expected impacts is the understanding of soil carbon sequestration that contributes to climate change mitigation, exactly the topic of the webinar today. And we identified that we had 4 categories of question. We needed to improve knowledge needs, to improve understanding of organic carbon sequestration, and we will have calls on the role of soil biota, on the permanence of soil carbon, on saturation, on deep soil carbon, on stoichiometry. We need to evaluate soil management options, and we are focusing on the effect of management options that allows compromises between soil organic carbon and other functions expected from soils. We focus, and that's the topic of today again, on identifying, predicting, and reducing trade-offs with nitrogen losses and greenhouse gas emissions. And so far, we have 3 research projects already financed that are working, 2 on mineral soils and 1 on organic soils, very different organic soils. And we want to quantify the potential of organic carbon sequestration. We have 1 project doing that on 23 European countries. And we want to do scenario modeling. We think it's necessary to do scenario modeling with changes in policy, changes in climate, changes in consumer habits. What is also needed in terms of knowledge in this area is, of course, to develop measuring, reporting, and verifying organic carbon sequestration. So we work at present on remote sensing, and we'll work on proximal sensing. And we do not have yet projects on that, but we think that there are a lot of knowledge needs regarding the enabling conditions for enhanced soil organic carbon sequestration. And on that, I would like to thank you for your attention.
Wonderful. Thank you very much, Claire. I think that was a a terrific broad overview of the issues surrounding achieving net greenhouse gas removal in soil, and the case studies were really fascinating too. I now have some papers I have to go read, clearly. So I'm going to address some questions that came up from the audience today, and then also that were submitted during the registration process. And one question that came up more than once that I think is an interesting one is how, apart from different management techniques that apply within an annual cropping system, how does shifting to more perennial land cover affect N2O in particular? And do we know anything about this?
Regarding N2O, there has been, I'm thinking especially into, well, depends on the fertilization of your perennials. Well, the main factor of N2O emissions is the application of nitrogen fertilization to soils. I'm thinking of agroforestry. There has been some estimates of N2O emissions from agroforestry. In general, it is reduced compared to the counterpart. Well, there's no additional N2O emissions because, of course, the trees uptake the soil nitrogen, and they can uptake it even from very deep soil layers. Quite often also soils are drier, so conditions are not favorable for denitrification. But the only exception, there were some measurements with legume trees. And in that case, there could be additional N2O emissions.
Interesting, that makes sense. So another question that came up repeatedly in different forms that I think would be interesting to discuss is it relates to scaling. And so the field experiment that you were discussing involved this very, I imagine, very laborious set of chamber measurements to get N2O fluxes from one place. And then the modeling study covered vast areas. What do you think the right balance is of those sorts of approaches for doing large-scale monitoring of greenhouse gas emissions?
Difficult question.
Right.
Well,
maybe there isn't a right answer, but yeah, I'd still like to hear your thoughts on it.
Well, and I think, but you gave partly the answer, that we need to combine case studies, monitoring systems, and it's an issue to establish monitoring systems where not only where diverse measurements are made over time. These monitoring systems can be at long-term, coupled with long-term experiments, or just sites that are taken along a grid, like it is done in several countries, or at farmers' place. Well, deliberately at farmers' place with selected management options. So monitoring systems are needed, case studies are needed, and modeling helps to change spatial scales and time scales, I think. So yes, I do not have sorry, but I do not have the good answer to your question. What yes, one thing I would like to say is that, as it's in another area, as an editor, I am in a journal, I am amazed at how we tend to undervalue case studies. But to do a meta-analysis, you need case studies. To calibrate, to validate a model, you need case studies. So even though as scientists we tend to undervalue case studies, we absolutely need them. But maybe we need to set them in a more coordinated way, that is along a framework. Where do we lack information? For which, pedoclimatic versus management conditions do we lack information? So maybe the effort should be done in that direction.
Yeah, that's a very good thought. Thank you for that insight. I think that's important for everyone to remember, because we tend to go to the meta-analyses when we want big picture answers, but we can't always answer our questions at that scale. So, all right, well, we're getting close to the halfway mark here, so we'll transition to our next speaker. And before we do, I forgot to mention something at the start. Firstly, feel free to post questions at any time in the questions feature, but I should also mention that we have a discussion forum we've set up on the ISCN website via this Padlet platform. And so if you go to the chat, then attendees are welcome to post further questions and discuss things amongst themselves on the Padlet even after the webinar is done. So take a look at that if you're interested in continuing the discussion. And without further ado, we'll move on to hearing Jane Johnson.
Thank you, Eric. Want to give me I think things are working. So if not, somebody please let me know.
Yep,
Claire, you did a fabulous job of setting me up. I think our talks are going to complement very well. Again, I'm Jane Johnson. I'm the research leader and a research soil scientist with USDA-ARS in Morse, Minnesota. This is a corn soybean system that is primarily, we're in the middle of well, not the middle of the corn belt, but certainly part of the corn belt. And so Claire set us up very nicely, and if we think about this question and we're trying to get at how to answer this idea of the carbon stocks, how are the carbon stocks changing, and how do we look at that in terms of net carbon emissions? And that has to include the other greenhouse gases, particularly nitrous oxide. And if you're in animal systems, then it also becomes important to be looking at methane as well. So this is out of the recent, fairly recent state of the carbon cycle report that came out in 2018. And really this kind of drives home the importance of looking at the fact that there's management drivers from agriculture that could be soil amendments. Claire did a nice job of talking about lateral movement when you use manures or other things. Soil management, tillage, etc., the production intensity. So these are all important management drivers. They interact then with the climate change, annual weather, the extremes. Those are all going to have a role on these dynamic processes. Also looking at technology drivers. Are we improving our ability to look at nutrient efficiency, whether it's slow release fertilizers, are they actually changing the amount of nitrous oxide that comes off, or do they just simply change the timing? And then the role of the microbial community. The soil microbiome is a huge area of research from a vast variety of research questions, and then looking at production, etc. Also overlaying this is policy and then the social and economic drivers, which are well beyond my expertise, but they certainly have an important role to play as well as we move forward. So if we think about some of the mitigation strategies that from a farming community you may have at your disposal, you have things like crop rotation and complexity, going from continuous corn all the way up to very long rotations that may be 7 or 8 years in length, and what they might do with nitrous oxide, and that can be, they might reduce or decrease the frequency of high demand crops, which could reduce the amount of nitrous oxide and provide additional benefits. But again, you have to go in and look at it very carefully, just as Claire had shown in her studies. Cover crops, part of it depends on what are the cover crops. Are they legumes? Are they winter kill, et cetera, et cetera? And there's a limited number of studies that have really dug into the effect of cover crops that may be going on. You have tillage, and Claire did a wonderful job of showing, some of the issues with tillage is that you have to make sure that you're not just redistributing it. And it changes the microclimate and changes the growing conditions that may be happening there. So it's important to understand what's really going on there and looking at least one increment below the greatest step, the tillage, and seeing what's really happening over time and within those treatments. The timing of nitrogen becomes important. and then also looking at perennials. And I'll give a couple case studies from some research that I've done in Morse that addresses some of these options. The question came up about scale, and that it depends on the question you're trying to answer. For one thing, are you trying to do national or regional accounting, or are you trying to answer something that's more specific at a regional or ecosystem? Most of the research I've done has been at the field scale, where we go out and we poke holes in the ground, we collect those soil samples, we take them back to the lab, and we try to account for temporal and spatial variability. Both of them are certainly can be an issue. You're looking for small changes against a large background. Knowing where you start is very important. I'd encourage you, if you're interested in methodology, there's a recent paper that came out by Nyack et al. That does a very nice job of reviewing, some of the options that may be available. Likewise, with nitrous oxide, there's a variety of measuring techniques. Probably the one that has gotten the most leverages or the most papers put out are the simple chambers, closed vented chambers. It can be manual or automated. They have the advantage is you can put a lot of them out in the field to try to capture some of the spatial variability. They are hampered by temporal variability. If you use automated chambers, you tend to get better temporal variability captured at the expense of the spatial variability. There's new micrometeorological methods coming online. Somewhere along the line, usually have electron capture as one of the tools for measuring by chromatography. There's some optical methods. And again, there's an analysis paper on the methods that came out in 2014. I'd encourage, if this is a topic of interest, to check it out. For the studies that we did in Morse, typically we were going out and collecting. We'd pull a sample, we'd transfer that into a vial, take it back to the laboratory. We measured these samples year round. We'd go from planting to planting. Minnesota has very cold winters, and we tend to get a pretty substantial spring flux of nitrous oxide that comes out. We did look at methane, but in these row crop systems, typically we've seen very little methane uptake or emission. They tended to be very close to backgrounds, so typically we've neglected, going out and measuring them. Like I said, if you were having systems where you're looking at livestock, then methane becomes way more important. Or if you're looking at rice systems, for example, then you would have to pay more attention to the methane. So we would go out, we would take repeated measures. So we'd come back to the same spot, take another sample after these chambers were put on, calculate the flux, and then integrate by triangulation among time to get our integrated point. These great job, I think, of capturing some spatial variability hampered by temporal variability. Once you have the emissions, you can convert that then by CO2 equivalents, as was previously mentioned, to account for the global warming potential. The values there that are recommended have changed over time. And for nitrous oxide, typically they've reduced the value on a 100-year basis, but increased methane. Nonetheless, you have to make that calculation if you're going to compare apples to apples. So I'm going to kind of transition and talk about just a couple of examples that we've looked at in Morse in terms of what has been, some of the studies, these go back, over the decade or so. So one of the first studies that I did on nitrous oxide was part of a group effort referred to as GRACENET, which stands for Greenhouse Gas Reduction through Agricultural Carbon Enhancement. And it was an effort that was spearheaded back in the early 2000s by ARS scientists, really trying to start getting a handle on what was going on in agriculture in terms of carbon sequestration. Was it being sequestered? How can we store more carbon? And we had delightful debates about what and how to measure. So in this particular study, we selected 3 scenarios. What was considered business as usual. So in Minnesota, that was a corn-soybean rotation. Inorganic fertilizer, probably at that time, I believe it was probably anhydrous ammonia, and then intensive tillage. We also had a maximum carbon sequestration treatment, which was a longer rotation. The only time it was fertilized was during the wheat phase. The corn relied on the nitrogen from the legumes in the system with reduced tillage. And finally, this idea of an optimum greenhouse gas benefit where there was no fertilizer applied. So the fertilizer or the nitrogen for the non-legumes had to come from the previous legumes in the system. The idea being if fertilizer is a source of nitrous oxide, that should reduce it, and the longer rotation should be increasing the carbon. So if we sum this and take a look at it, this work has been published some time ago. But one of the things you have to be careful of with carbon dioxide is that in these 2 systems, because we had wheat and alfalfa, as soon as you put that chamber down, you're going to start getting erroneous CO2 measurements. It's going to overestimate the amount of CO2 coming off compared to the business as usual, because you can put the chambers between those row crops rather than over it. As I had mentioned, the methane is very, very low. This has already been converted to mega equivalents, and it's incredibly close to the detection limit of our equipment. So these are very small values and quite negligible when you look at, the multiple differences here. So methane, we would have predicted that this would be the lowest treatment, but when in reality it didn't matter what the source of that nitrogen was coming from, we still had about the same amount of nitrous oxide that came off on all 3 of these systems. We also measured the change in carbons. We had a baseline carbon. We knew had measured the bulk density, so we could calculate on a excuse me, calculate on a mass balance. So we needed to look at the change in carbon over time and not just among the treatments. So we were able to detect increases in carbon in all 3 systems, at least at the surface, but decreases at depth, so that overall the greatest loss was detected in the business as usual, but also some evidence that we may have been losing some carbon in the other treatments as well, although statistically this is not different from 0. Similar to the graphs that Claire had shown, if we sum what's going on, so we have production inputs, that would include things like the fuel that was put on and the fertilizer that were calculated. Those are the black bars here. The methane is so small it gets lost in translation in the graph. Here's the nitrous oxide and then the loss in carbon. So in all 3 of these systems, we were losing, we were going backwards. All of them were net sources of greenhouse gas, and we just were not seeing enough carbon storage to see a difference. Now, if we kept these going for another decade, considering how slow things change, would we? Maybe. We could speculate that that may happen eventually. But nonetheless, we didn't see 0, but at least it was less. So I guess that's a positive, sort of. Not going to get us to net 0, though. In another study that we did that on a similar type of system, the plots I was talking about before were over here. Now we shifted. These plots were established in 2000. They had been in no-till since then. It's a slightly wetter area on the field. Again, we have corn and soybean as our baseline system. Here's the chambers. And alongside we have perennial grasses. These were being managed for biofuels. So these were intensely managed grasses. They were not trying to mimic a native system. They were looking at this from the context of bioenergy, which ideally bioenergy would shift to biogenic use of carbon rather than fossil carbon, but you have to be careful not to cause problems with your soil organic material as well. So the big blue stem or the switchgrass would be harvested either in the fall or in the spring. And then we monitored the change in carbon. We had baseline numbers. We had 2006, which was done shortly before we started measuring the greenhouse gases, and then inclusive of a fall sample in 2011. And we measured greenhouse gas for 3 years. This work has been published in SSSAJ, Soil Science Society of America Journal in 2016. So this gives you a nice example of the kind of messy stuff you get when you go out in the field and you try to measure nitrous oxide with closed vented chambers. You spend a lot of time out in the field and you come back, you bring them in, and you're at or near the detection limit, interrupted by spikes that are caused either by fertilizer events, which are signified by the gray arrows across the top. And the other event pointed out here with the blue arrows are spring thaws. Now this is, as I mentioned, we're in a northern climate. It gets very cold. We tend to have snowfall. And in the spring, sometimes before you have true flux in terms of total thawing, you get the soil surface thawing, you have disruption of the cell contents of anything may have been there, you have nitrogen available, you have soluble carbon, and you get these spikes in nitrous oxide coming off. Interesting though, we didn't see that in our perennial grasses. We see the impact of the fertilizer. Like I said, these were intensely managed, so they were fertilized. And depending on the climatic conditions, the weather conditions of the time, the spike from the fertilizer could be quite variable. If it was dry at fertilization, you don't see denitrification and pretty limited amount of nitrous oxide coming off those systems. We also measured the change in carbon. So the carbon that was stored under the grasses, again, these were managed, so that residue was being harvested every year. So we're not seeing a huge buildup in the amount of soil organic carbon that was there. We had some storage, particularly significant at the surface under the grasses, but not under the corn and soybeans. The star means that it was a significant difference based on the regression line for those. So from time 1 to time 3 and regressing that data. So if we look at those, again, a negative offset implies a reduction in the global warming potential. If this is real, then perhaps we are getting an offset, but the difference in the change in carbon was not significantly different from 0, although it is encouraging to see it's at least going in the right direction. Perennial grasses did not guarantee that this was going to happen. Depending on when we harvested, there seemed to be an influence of the management of those grasses, and the corn and soybean certainly was the greatest emitter. Even though this isn't a significant difference, it does not bode well, and that is, I guess, as predicted. Even though it's under no-till, we're still having a positive influence. In terms of the nitrogen management on these switchgrass treatments, there is an opportunity here that I think we can do some improved management with the nitrogen fertilizer on those crops to improve those numbers. So if you're trying to really get a handle at what's going on in SOC and greenhouse gas, you have to deal with the fact of temporal and spatial variability. And that's true for the nitrous oxide, it's true for the soil organic carbon. And accept the fact that you're going to have trade-offs. If you're trying to really delve into good temporal data, you may do that at the risk or at the expense of spatial values. The new nitrous oxide sensors that are coming online that we're hoping to install yet this year will get much better temporal values on what's going on with nitrous oxide, but it will be on one farmer's field and one cropping system. So alone, it's a case study, but it will have to be coupled then to other studies across the region using this type of technology. And what works well here may not work as well someplace else. Bulk density is highly variable. The depth is important. And there's a lot of ongoing research through ARS, university partners, and in the international community. I just want to leave you with just a list of some of the resources. When I look through some of the questions that Claire Phillips had shared that people had submitted, I was impressed by the depth and the extent. There are some incredible resources that are publicly available, including the work that's done by EPA. I have ARS scientists that help with the modeling components of that for trying to get a handle on what's going on our greenhouse gas inventory on an annual basis. The Soccer 2 report included over 200 authors from the United States, Canada, and Mexico, went through very intensive review and internal and external review. I'd like to bring to your attention the USDA-ARS Agricultural Collaborative Research and Outcome System, AgCross. If you're looking to just get a feel for where the studies are in the United States, it's a great opportunity. A lot of data that's housed there. The relatively new, although it's been around now for about 5 years, is the USDA Agricultural Long-Term Agroecosystems Research Network. And they're really trying to get a handle on what is sustainable. How do we balance the inputs? And it's looking at row crops, it's looking at grazing systems. So it's very broad and figuring out ways how to bring this data together. During my introduction, they had mentioned the Global Research Alliance on Agricultural Carbon or Agricultural Greenhouse Gases. Last I looked, I believe they're up to over 90 participating countries. It also has a great set of resources on how to, some training videos that are available. So I'd encourage checking it out. If there's a group the working group on livestock, there's a working group on cropping systems, paddy rice, with really phenomenal research and opportunities for the cropping systems, and I believe it may extend into the livestock. There's Magnet, which houses the metadata for many of the studies that are available to help people get an idea of who's out there doing the work. And of course, then the IPCC, who's been around now for a good long time. They just came out with the new annual new report, number 6. And I know it's gotten some media play, and I have not had a chance to really go in and dig into it. But again, an incredible resource for those that are interested in climate change and trying to understand how do we find these answers. Earlier this spring or this year, I was part of a network orchestrated and to discuss how do we achieve net zero. And there's what research questions can ARS and our partners help answer? So it is a very timely subject, and I applaud the organizers for putting this together and very excited to see the number of people that are attending. And with that, I'll turn it back over to you, Eric, to cover any questions you may have.
Thanks, Jane. That was terrific. It was great to delve into some of those case studies that Claire was mentioning earlier, and to see the complexity of the different outcomes in these systems. Often things don't seem to respond the way we expect, or that we hope, perhaps. Let's see. In terms of questions, one question that came up. This is almost a philosophical question more than it is a scientific one, perhaps, but depends on how you look at it. Global warming potentials are referenced to different timeframes. This is something you mentioned earlier on in your talk. So what does it mean to pick a particular time frame for calculating global warming potentials? And do you have thoughts about whether a 30-year versus a 100-year time frame is more appropriate? And what do we mean when we communicate that way by choosing a temporal reference point?
I'm not 100% sure. I do know that the most common one I see is 100 years. And I mean, 30 years makes sense because we can understand what may happen and we can think about, we maybe can get our hands around 30 years. 100 years seems like a long time. Most of us are not going to make it for 100 years. But I think if you want to have extended change, then the 100 years makes sense. And if you dig into the IPC and they do have a chapter that explains, the rationale behind this, how those numbers come in, and some gases don't last long. They have a very short lifetime. CO2 has a very long lifetime in the atmosphere, which is one of the reasons it's used as the benchmark. Nitrous oxide has a fairly long compared to methane, which is shorter, but it also is constantly being produced. So I think it, again, it comes back to what is the question you're trying to answer?
Great, thanks. So another question that I was thinking about, and that relates to some of the other questions we received, is about climate. At various points, you talked about the thaw events and the N2O related to those thaw events. And I'm wondering, are there particular climate domains that are more susceptible to N2O emissions, say snow-dominated climates, for instance? Do you imagine that the concerns might be different in warmer climates farther to the south?
Oh, absolutely. There's the spring thaw event really is a phenomenon of soils that freeze and thaw. I have colleagues that, were, in Indiana, for example, and even though they were out there looking for those events, they didn't happen. There's work that's come out of Germany where they've seen it and some other northern climates. So if you have these thawing or freezing cycles where the soil is frozen at depth and then it thaws back out, you're more apt to see it. If you have very mild winters, there is no true thawing event that happens. And kind of colloquially, what I envision is happening is that soil's frozen. And if I was going to predict today to see nitrous oxide coming off of a field in a northern climate like Minnesota, it would probably be mid to late February on one of those days where the sun is out and intense, and you get this slimy slurry about a half an inch or a centimeter in depth that's thawed. And so the water can't go anywhere. It's denitrifying, it's cold. The only place for those microbes are, they're awake. It's warm enough for them to go. And so they're chewing up whatever's available and it's denitrifying. So you have all this nitrous oxide come off and it freezes again at night and everything stops. And the next day it may happen again. So it's very ephemeral, but it can be 30 to 50% of the nitrous oxide we've measured out in those fields for the course of the year. So it should not be neglected. It's difficult to model because it's so ephemeral. It's difficult to measure. So I'm excited when we get this new sensors coming in out at the field to really get a better handle on what might be coming, even though it is a one spot, but we will be able to look more closely at those temporal variabilities. Good question. I like that one.
Yeah, it's very interesting. I even, just a brief follow-up question is there, given that it's an ephemeral event, perhaps that's encouraging because it gives us something to target, right? But are there management approaches that could target that brief window? Or is it really out of our control because the soil's going to thaw, and if there's nitrogen available, what will happen will happen? Is there a way to prepare for that event?
It really is. I don't know that there's a way to prepare for it per se. If you have a source of nitrogen in that soil and it freezes, you're going to have some nitrous oxide coming off. It does come back to the importance of making sure that you, and some of it might fall into this indirect. I mean, if the nitrogen didn't get used up, it's sitting there in the materials on the soil, and then you get this freeze thaw event, you have a source of nitrogen, you have a source of carbon. What is intriguing is that when we looked at it in those perennial systems with the grasses, we did not see the same flux that happened. They're covered, they're protected, they're free. It would be an interesting study to very carefully go in and measure those temperature gradients that may be happening under those systems compared to a more bare soil. We know that the microclimate, for example, is going to be quite different under those perennial systems than what they are, and that would be a grassy system rather than a legume compared to, you know, a row crop. So I think there's lots of research questions still available.
Great. All right, well, we've reached the hour. Once again, thank you to both of our speakers. This has been a fascinating webinar, and anybody that still has questions or topics they'd like to discuss can go to the ISCN website and find the Padlet discussion forum there. Thank you all, and we'll see you next month to hear about tillage.
Excellent, thank you.
Thank you.